Good morning, everybody. We're about to start our day 2 workshop, so please be seated. Good morning, everyone. Good evening, or good afternoon or good evening if you are joining remotely. Before we start another exciting day, for the second day of this workshop, I will just take a few minutes to do a quick summary of our day 1 in case you missed day 1's presentations or you were online. So we can, this is an opportunity for me to summarize what we discussed in person. So for the talks, yesterday we really had an eventful, so I'll break it from the FDA side and external. So from the FDA side, if you are interested to really have a quick overview of what has happened in the past 10 years, how the generic ophthalmic products evolved, and what kind of research and outcomes, PSG development, current triage process, please listen to Andre's talk. He has done a really good job summarizing all those information in his presentation. And then Bruce really did an excellent job comparing the traditional, more conservative approaches for the agency assessing the ophthalmic generics versus our updated thinking on how we would treat, for instance, for simple solutions, if you have permissible formulation differences, what would be the most efficient approach, or what are the alternatives to the previous default as warranting in vivo BE study. Then follow along the line, Yuruki's talk provided 4 case studies about how companies are trying to request BioWaiver when they have formulation differences in the exception excipients. There are 2 failed cases. Those cases are not necessarily seeing those differences always would still warrant in vivo study, especially per our current paradigm. But those are really emphasize, if you do have those differences, right, you have to provide very strong scientific justification, and it's highly encouraged you talk to us pre-submission during your development so we can give you constructive feedback. Then move on to Xiaomi's talk about the innovative IVRT methods. We know not just IVRT, but in general, when FDA look into any modern technology or do anything appear to be innovative, I think it's a mixed feeling at the company side, and often they also panic, feel like, is that something we are expected to do? So I think the key message in that is not necessarily, as Xiaomi has emphasized in his talk, what we're trying to convey there is we do appreciate and encourage innovation, but those studies for the FDA to conduct, we have also multiple purposes. Like for the studies, the lab was able to really get to the bottom of the release mechanism of those complex products. Those understanding is beneficial for company to really guide your development. And those methods eventually, hopefully, will be more adaptable to be supported as either one-time study or routine use. So the key message there is to not be afraid of innovation, trying to push and think more beyond the idea, not just the methodology, to look what that methodology brings to your knowledge. And similarly, sometimes by doing those studies, then we really get a fundamental understanding of the product. So then we can make a clear determination. Certain things are not necessary, or certain differences do not lead to meaningful differences. So do not always panic when you see FDA is innovative and doing something novel. Then if you are interested in learning what are the most commonly seen deficiencies for ophthalmic products assessment, listen to Hee-sung's presentation. She has a very comprehensive summary there. We know there is some contradiction in terms of Xiaomi presented a talk about using non-sync condition to do the IVRT, and he's listed sync condition or not being sync condition as one of the common deficiencies. Then again, there, I think Skin condition is still, most cases, a gold standard, but then again, it's a scientific development and a scientific assessment. So if you do think non-skin condition give you advantage to make your method more biorelevant or discriminatory, more discriminatory, then provide that justification and discussion to make a case. We will not just simply reject a method because it's non-skin condition. That's not the case. So I want to make that clear because there is appeared contradictory, messages there. Then move on to Ming Liang's talk. He really gave a very encouraging presentation about how modeling may play a role and how we may gradually step into conducting virtual BE. So those are still ongoing effort, and certainly it is still at early stage, but significant progress has been made. So switch, I hope I didn't miss anyone from the FDA side. And then switch to industry side. I think Sandoz case is highly appreciated, and it's really, I feel like it complemented Bruce's talk. So if you experience similar struggles as Sandoz presented in their presentation, please listen to Bruce's presentation, because those are, also read our PSGs. I know sometimes, for the simple solution for ophthalmic product, at least we publish PSGs for every single one, including solution products for a reason. And then when you read the PSG, also read the whole PSG, including footnote. Don't skip, because sometimes those things, I know you are busy and you read through, but those are important messages and reflect our current thinking. Then Dr. Atto, Professor Atto's talk on the preservative is related to Bruce's talk and also somewhat related to Euroke's and Sandoz. And one motivation for us to conduct that research is really to understand those struggles, right? We have literature indicating change of preservative could impact local bioavailability, but is that always the case? Is that true? To what extent? Those questions are FDA's effort really trying to move things forward, to step away from the conventional conservative approach. But we do need data, because at the end of the day, all our PSGs are really evidence science-based. So those things take time, but that's where we are working on. And we also highly encourage, if company, you have similar data, share with us, make your case. Don't just always feel like you may get rejected. Yes, you may get rejected, but if you don't try, you for sure will not move forward. Then I think Sloris talk really coupled well with Professor Shuling Du's talk on the ointment product. I think we have made a lot of progress in terms of particle size measurement. We had an excellent workshop back in September. Again, if you missed that, please listen to the recording. You will find it very helpful. But for ointment, those semi-solids, it seems like particle size measurement is still a challenge. That's where we conducted our research and also IVRT of those dosage forms. So really the concerns were heard, noted, and we will continue looking to those areas. Then move on to the last part. We had actually 3 talks, 2 external modeling talk and also a PK/PD animal data share from 3 external presentations, and those mostly focused on bronzonamide ophthalmic suspension product. And we are aware IOP suspension product have been companies struggle, and there are lots of interest in developing, but the current recommendation for comparative clinical endpoint studies have its own unique challenges. So those things we are aware, and the modeling work certainly had played a critical role to update our current understanding and move us more close towards what we're trying to achieve in terms of understand the critical quality attributes impact to clinical relevance to really make us confident if we develop an alternative BE approach. But a little bit more background to share on that line is for modeling, I think we talked about in the in-person group, small group as well, is often big company or everybody struggle with data. So when we first trying to use modeling, we struggle with data too. There is just not enough data for us to develop the model. So actually all the data presented yesterday in the modeling presentation, all the animal, those formulations were made by our FDA lab folks. It took us 2 years with multiple people to manufacture those 5 different formulations, and it's very challenging to develop. Suspension formulations, we know that is not simple. So those work is really tremendous, and it takes time. So sometimes I feel the behind-the-scene work is not fully aware by company, and we appreciate your feedback, and we know you want us to move faster, and we are trying our best to move fast, but you are developing products, so you know everything takes time. And we develop our own formulations to make solid science, and so we share some of your pain during development, and we work with our internal, external experts to really trying to move things forward. That really lead to additional discussion in the small group is another struggle to a certain extent is the Q1, Q2 for ophthalmic products. Today you will hear more. For instance, we are aware when for polymeric excipients is never a clean cut. How do you really define Q1 being the same? Then on top of that, if those polymeric systems are cross-linked, they lose identity after manufacturing. Reverse engineering can be very challenging. How you achieve Q2? It's good to hear those pinpoints from you because it's assurance like we are on the right track. So today you will hear talk on those points, and we discussed some of those yesterday in person. And then for formulation characterization and in vitro drug release testing development, again, it's really appreciative to hear all your honest feedback, and to a certain extent, that's encouraging. And those are difficult questions, but I feel like it's important to talk about them. And we acknowledge and we are aware, I would say, for most of those. But sometimes, like I mentioned, we are not in where you are, so we don't maybe always know exact practical struggles you may face in your daily development, and that's why we host this workshop to solicit those to help us move things forward. So with that, I really want to thank everybody's contribution again. From yesterday, our speakers, our in-person attendee, and our virtual attendee. And I'm sure you will find today, again, being exciting and meaningful. So with that, I'll pass the podium for Dr. William Smith to kick off session 4. Thank you.
Hello, everybody. It is my pleasure to kick off session 4. So this is related to insertable and implantable products. Where is my little clicker? There it is. I'm Dr. William Smith. I'm a research scientist in the Office of Pharmaceutical Quality Research. So today we have a number of really nice speakers, talks as far as you think back to yesterday, Andre's talk, he showed kind of that difference between the gap in what products we still don't have generics for. And over the last 10 years, we went from not having a lot in the space of emulsions and those kind of things. And this is still one of the big areas where there are still gaps. So this is kind of the front moving edge of a lot of this kind of regulatory science on these products. So today, We have with us Dr. Michael VandenBerg. He's a chemical engineer in also the Division of Pharmaceutical Quality Research at the FDA. We also have a postdoctoral research fellow, Coleman Johnson, recently from UT Austin, who will be sharing some of his work that he is continuing on in the Office of Pharmaceutical Quality Research. We will have Dr. Kinam Park, who will be joining us virtually to give his talk. He is a professor at Purdue and also the president of Akina Inc. We also have Andrew Clark, who is the Senior Director of Research and Strategy at DigiM Solutions, which will be a really fun talk. And we have, last but not least, Megha Barot. She's a pharmaceutical scientist in the Office of Product Quality Assurance to kind of give a regulatory perspective on everything. And with that, I'm going to welcome up Dr. Mike VandenBerg for the first talk.
All right, well, good morning to all of those in the room with me, and good afternoon and good evening to those online. It's my privilege to be able to share with you our work on dexamethasone intracanalicular inserts and the research we've done at OPQR. So yeah, I am, like Billy introduced, thank you, Billy. I'm a chemical engineer in the Office of Pharmaceutical Quality Research. So this is our talk on product evaluation, performance characterization of dexamethasone intracanalicular inserts. So my disclaimer, this presentation reflects my views and not the views of the agency or its policies. And I included this slide because I like it, and I think it reminds us of like what we're all doing here is very important. And as Markham mentioned in his talk yesterday, this affects millions of people and their quality of life, especially for ophthalmics. So it's very important work that we're doing. So a lot of talks yesterday covered this in great detail, but I just want to reiterate some of these points again. There's significant challenges with BE establishment for ocular and ophthalmic products, especially for implantables. So for in vivo studies for these products, bioequivalence studies with PK endpoints can be quite challenging, considering that conventional PK studies that measure drug concentration in plasma generally totally infeasible due to negligible systemic exposure from the drug release of these products. And then it can be difficult or practically infeasible to conduct an in vivo aqueous humor PK study. And many times these are prolonged studies that add additional hurdles to accomplishing, collecting good data and having a robust study. And then furthermore, doing comparative bioequivalence studies with clinical endpoints, again, it's a very long study and there's ethical concerns. And then there's careful considerations of how you include a control arm here, and then the sensitivity of the study, looking at the different outcomes and using that to establish bioequivalence of the products can be challenging due to the low sensitivity. So as alternatives to in vivo studies, there's been increasing attention to using in vitro studies as a surrogate or in silico modeling to assist this, to be able to establish bioequivalence and look at sameness of these types of products this way instead. So for in vitro studies, requires comprehensive understanding on how the CQAs and also the manufacturing process parameters of the drug product can affect the in vivo performance. And then oftentimes this also requires resolving very difficult analytical challenges. So like we've talked about quite a bit so far, the Q1, Q2 determination for cross-linked polymers, for example, is very challenging. And we have to evaluate if these types of studies can be done in vitro and if it's feasible to look at these types of products and compare them this way. So in developing a regulatory stance and pathway, as well as establishing the scientific foundation and rationale that underpins these, there's quite a number of challenges involved. And so first of all, established in Q1, Q2 sameness, as I've already mentioned, brings up questions such as, so how variable are non-compendial polymers that are sourced from different vendors? Also, along with that, can the finished product even be feasibly reverse engineered? what does Q1, Q2 sameness for a covalently cross-linked product look like? As Jhon mentioned, you have a mixture of things that go in the pot, and maybe you can establish Q1, Q2 for those, but after you have this irreversible cross-linking, what is on the other side of that? What's Q1, Q2 then? For physical and structural equivalence for these types of implants and inserts also, what are the key critical quality attributes of this drug product that you have to match to get the same kind of therapeutic performance? How does the processing of them and how you manufacture these affect the microstructure? And then in turn, how does that microstructure affect product performance? So they're Q3 considerations. A lot of these products they swell, they're hydrogels, and they oftentimes biodegrade, and there's concerns that come with this. So can the drug release, for instance, be measured comparatively in a meaningful way and be discriminative against product Q1, Q2, Q3 differences? And then how do you conduct a good IVRT for example, an intracanalicular insert, which we'll be talking about? And then how to comparatively measure the swelling of these products and the degradation in vitro, and how to connect that to what would happen in vivo. And so there's comprehensive characterization needs, and what are the comparative studies that need to be done to ensure performance sameness. So these are some of the questions that OGD, ORS, our colleagues there face regularly. And they have a collaborative approach where they often reach out to us in the research division to help them basically understand the science that underlies some of these issues to guide decision-making. So we have as was mentioned yesterday, we have a subject matter expert triage team process where when an NDA is approved, basically the offices come together and we deliberate and discuss if there's research needed to be able to draft a PSG, to be able to facilitate the development of generic version of these types of products, which are often complex products. So for intracanalicular implants, this is an example. This particular product is a dexamethasone intracanalicular implant. This is, as you can see in the diagram on the left, these are placed through the lower punctum, which is about a 0.5 millimeter wide opening near the inner part of the eye. And this goes into the canaliculus, which is basically the tear duct. And it sits in the vertical part of the canaliculus there, and it swells to basically expand and then fit snugly in place there to remain there as a punctum plug. And it remains swollen and gradually releases the entrapped drug API. In the case that we're going to be talking about would be dexamethasone, which is poorly water soluble. So this leads to extended prolonged release duration. This is accomplished by using a covalently cross-linked hydrogel in the case of the commercial product. And it uses this 4-arm polyethylene glycol that has terminal end groups that can react with amines and cause an amidation reaction for the cross-linking. So you can see that the insert is initially a smaller radius. It's placed into the punctum and into the canaliculus. It swells with a known swelling kinetics, and then it expands its diameter so that it basically matches it's near the diameter of the canaliculus. So there's product-specific considerations for this particular product. So the known drug product characteristics that are relevant to the performance here, among them are, this is an intracanalicular insert for ocular inflammation, in the case of a dexamethasone insert, and it needs to be designed for easy implantation by a practitioner. The dry diameter of the insert matches the average punctum diameter for an optimal fit. This can introduce challenges in vivo studies again, because the real punctum diameters and canaliculus diameters have pretty wide range in patients. It also needs to provide continuous API release for improved compliance versus eye drops, which need to be regularly administered to a patient, and this causes problems with patients forgetting or not applying it correctly, et cetera. It needs to swell radially upon implantation. This ensures secure placement so that it doesn't pop out or fall deeper into the canaliculus. It needs to be optimal in terms of the swelling and the dimensions pre and post swelling. and it functions as a punctum plug. It resides there at the site of action. The tear fluid swells it and gradually basically courses through the hydrogel and absorbs the dexamethasone that's entrapped within it, and it bathes the ocular tissues with the very low concentration dexamethasone fluid. And then oftentimes, in this case, it contains a dye that's used as a visual aid. This helps the practitioner to verify that it's retained at the site of interest. And then it contains a crystalline API that's homogeneously dispersed in the polymeric matrix in particulate form. And the drug release happens over a long duration, from weeks to months, mainly through diffusion into the tear fluid. And then finally, these are resorbable inserts that don't need an active procedure to remove. They'll just resorb and degrade over time. It's not expected that they remain in the canaliculus beyond a couple months, depending on the chemistry that's used to make the cross-linked hydrogel. So for a generic dexamethasone ophthalmic insert, there's basically some key properties there that would have to be considered when evaluating sameness. The drug particle size distribution and spatial distribution within the insert itself is important. The insert matrix, so in terms of the identity, so Q2, Q1 aspect of that, as well as the cross-linking density that ends up happening after the curing, the swelling and the degradation behavior that the insert features, as well as the drug release behavior. And so this, ultimately, all of this leads to an approach that's kind of like fingerprinting and requires a totality of evidence type of approach, where you look at all these different orthogonal aspects of the product to gain a comprehensive understanding when it can be difficult to measure these in other direct ways. So looking at the cross-linking mechanism and manufacturing that's used to produce the implant. So this is the chemistry behind it in this particular case. So there's a solution that contains dexamethasone, water, and then sodium phosphate buffer, as well as the one key functional excipient, which is the 4-arm 20K PEG with succinyl glutarate end groups. And this gets combined with the cross-linking reagent, which is this trilysine acetate, which contains these terminal amines. And this is also usually conjugated with a dye so that the insert can be visualized. And then this precedes a cross-linking reaction where amide bonds are formed, and you have basically you have a dispersed particles of dexamethasone within this covalently cross-linked hydrogel matrix, and then you end up with the free antigenous end groups that pop off the polymers. And then we come to the manufacturing process and how you make the implants. So first, there's a mixing step, and then you shape form the implant into the shape that you want, and then you have to dry or cool the implants to equilibrate them. And then from there, you do cutting and then QA and store them. So these are an example of the commercial insert, as well as some of the in-house inserts that we made. You can notice that in some cases we opted to exclude the dye, and we also made them at different dimensions to simplify the study and look at various different aspects of the process. And then from here, it leads to inserts that are able to swell and accomplish the features that we talked about. So the characterization of functional excipients is one of the first things that we did. So we took 3 different vendors that made the 4-arm 20K polymer named V1, V2, V3, and we also looked at an 8-arm variant of this and a 40K polymer variant of this. And this was in order to basically look at the differences in Q1 of these different polymers that had different properties. And these are all nominally the same polymers, but they actually, truly, when we measure them, have slightly different functionalization and molecular weight and purity. And so we use proton NMR, FTIR, and SEC, as well as MALDI, time of flight, to basically establish the functionalization percent, the molecular weight of the polymers, as well as the purity. And then putting those things together, we were able to come up with kind of an statistic for these that would describe the cross-linking capability of the polymers. And so this is the theoretical chemical cross-links on mass basis, which is those numbers in the bottom left. And you can see for V1, V2, they're quite similar. V3 was different than them by about 14%. And then the 8-arm and the 40K were kind of like control, or kind of like reference points to understand what if you use something that has double the cross-linking density, what if you use something that has double the molecular weight. So from here, we wanted to evaluate the mixing process and curing. And so using rheology on the pre-cured mixtures, as well as the mixture as it's gelling, you can do this, and you can look at the effects of buffer strength and discern the control over pH that's needed and what is crucial to control the curing kinetics, as well as giving you the ample enough window of mixing time that you need to manufacture this. You can look at the differences in the 4-arm and the 8-arm polymers, and you can see that these are relatively similar storage modulus, which is an interesting observation that we made here. And it points to the fact that there's physical cross-linking to consider, as well as geometric constraints and how the polymer molecules actually can all fit in space. And then when you have the dried finished inserts, there's different ways of measuring the mechanical properties of these. We employed dynamic mechanical analysis to look at the dried inserts, and we placed them in a tensile strain apparatus. We looked at the slope and strain response to get the elastic modulus, and the network stiffness here is a surrogate for the cross-link density, but it also tells you things about the pliability of this, the toughness, resistance to deformation, which are key properties that are important to applying these, because it tells you about how easy it is to break it or basically damage the surface of it, which is an important failure mode. And so the yield point also indicates processability, the different ways that you can manufacture it under different temperatures and conditions. And then it's also important here to consider the moisture content, which greatly affects the mechanical properties of the aerogels. And there's different ways of measuring it, including like compression, bending modes. And for these really small inserts, it's oftentimes difficult to do tensile experiments. So it's possible to do different types of mechanical testing. Okay, so as I mentioned before, the API is in a crystalline form, and also in the aerogel state, the polymers tend to crystallize, and you have like linear chain interactions. And so we can measure this and look at product differences using the different polymer types using DSC. And the melting behavior that we observe here, with the heat of melting and the temperature, the peak temperature and onset temperature of melting, we can learn things about the cross-linking density and the crystallinity. And this has forward implications having to do with the swelling rates and the underlying microstructure. And so we can see subtle differences between the different vendors that we used, with the V3 polymer that has poorer functionalization, for example, having a more similar thermogram to the 40K polymer, whereas the other vendors are more similar to the 8-arm. Using TGA, you can look at the suitability for doing something like hot melt extrusion to process these, and look at the degradation profiles under higher temperature. And you can notice here that the TLA is the most sensitive to that. And this can be used to measure the residual water content and thermal stability of the dried implant product. We wanted to look at the physical properties as well, the dry diameter and the density. We observed some differences in the 8-arm and the using non-micronized dexamethasone instead of micronized dexamethasone. We noticed that those caused a greater diameter on average post-processing. But between the other polymers, the 3 variants that we looked at, there was no substantial difference. There was actually a difference between the density between the V3 polymer, which had basically poorer functionalization than the other 2. And we noticed that this resulted in a slightly lower density. And this kind of comes back to the rheology and what we learned from there, that the viscosity of the pre-cured mixtures is likely impacting the mixing dynamics and the culpability to have air bubbles and pores form. And this results in a different density in the bulk state. And also looking at the pores, we can do XRM using our micro CT system. And so this is using X-ray imaging to look at the pore and the API distribution within the inserts. So this is very useful for telling the volume fraction of the pores, polymers, and API. And we can rank the porosity and the pore average size. And from there, we can also come up with the size distribution and the spatial distribution of these and compare how they relate to each other. And this is a very interesting way to get the in situ information about the API, like what's actually happening in the insert. And there's not many techniques that can do that. Besides this, you can maybe measure it before you do all of the manufacturing, but it's difficult to measure that otherwise within the finished insert. And then finally, the swelling is an important property that we've already talked about a lot. So looking at the shorter kinetics of swelling as well as the longer term, you want to have matching swelling kinetics in both of these regimes. And so we noticed some differences in the different polymers, but generally they all kind of trend the same, except you can see an obvious divergence with our V3 polymer there, which did have known impurities and it had poor cross-linking. And so it begins to degrade early and it swells well beyond the others. But this will come back into the release studies that we do as well. And then there's a strain memory effect where if you process these in a certain way by displacing them along a preferred axis, when you do the swelling later on, basically it won't swell very much in that dimension. And you can leverage this to make an insert that will swell in a preferred direction like we talked about with the radial swelling in the product so that it accomplished its therapeutic function in canaliculus. And by doing this kind of testing, you can figure out a sweet spot on how much change you want in the length and the diameter. And then finally, we did an IVRT method. And I know we've talked a lot about the controversies of different IVRT methodologies and how to make how to come up with an IVRT method that is best suited for the technique and discriminatory with respect to the key variables. Well, we used a static non-sync condition IVRT that we found was discriminatory with respect to the excipient selection as well as the API particle size, which is surprising. And in general, it followed the swelling trend quite well. So it supports that it's a matrix swelling mediated drug release mechanism. And it's slight divergence from Higuchi, which we talked about yesterday, incorporating a little bit of case 2 polymer relaxation, the swelling. And then looking at the degradation is important. And you can see from this chart, over the course of about a month, we have like a 10% decrease in most of the degradation. This is like the residual mass that's measured after leaving the implant in a water bath and then measuring the residual mass of the insert after it's degraded. And you can see that V3 is degrading rapidly even by like the 10, 15 day kind of area, whereas the others are kind of remaining steady. And in addition to this, you could do mechanical testing as well to verify the integrity of the network as the mass loss occurs. All right, so as we talked about at the beginning, establishing bioequivalence for ocular and ophthalmic products is pretty challenging. So we're working on in vitro options that are being increasingly explored through internal and external GDUFA research projects like this one. And here we looked at the CQAs of a complex ophthalmic insert product, and we tried to understand this product using a comprehensive suite of in vitro characterization techniques, trying to build out a toolkit that can be used to kind of characterize and fingerprint this product in a comprehensive way. This was used to gain deeper understanding of it and evaluate the in vitro characterization tools that are available that could potentially be used for demonstration of product sameness across variations in source material and manufacturing techniques. So regulatory science research serves to provide scientific support and rationale for FDA's PSG recommendations and review decisions, as well as share our findings and our outlook with the generic drug industry and broader public through publications, seminars, and workshops like this one. So these are references. You can come back and get this QR code if you want. But yeah, I encourage you to check out our paper if you're interested. And then I want to acknowledge my colleagues in OPQR, Rokan, Hussein, Christine, William, Billy, Zhao Ming, and then several others, as well as our colleagues in ORS, and the original triage team that planned out this project, and then our friends at DigiM, Lisa, who helped with the XRM imaging. Thank you.
Okay, next up, we have Dr. Kinam Park. Give us just a second. He is going to present virtually. So hopefully we'll get him online here in just a second.
I think I'm sharing slide now. Do you see that?
Not yet, but it's good to hear you. Oh, now we see him.
Right. Okay, good.
Go ahead.
Yeah. So first of all, I'm sorry that I'm not there in person, but I'm there in spirit. I know all of you. So anyway, today I'd like to talk about analytical method for mini-size formulation. And mini-size formulation is any formulation which have a total weight of 1 milligram or less. And small size make the assay a little bit difficult. So let me start with the currently available all long-acting formulations. They start with the oily formulations and aqueous suspension formulations. They are usually for very hydrophobic drugs and delivering large amount of monthly dose up to 1,000 milligram or 1 gram per month. But PLJ-based formulations are rather small in size and maximum dose for monthly administration is about less than 400 milligram, and it can deliver from very hydrophobic drugs to relatively very hydrophilic drugs to relatively hydrophobic drug because PLJ is the one to control the drug release kinetics. Currently, there are 27 FDA-approved PLJ-based formulations, and some of them are really complex. For example, sandostatin LAR is made of a glucose PLJ starchy polymer, which has been difficult to characterize, but only after FDA tried to figure out how to assay them, we developed the method. Also, Trelstar contains more than one type of PLGAs and also then makes the analysis a little bit difficult. But again, we were able to develop a new method because of the FDA initiative. Now, Ozurdex and Duristar are two ophthalmic formulations, and they are presented another difficulty because of their small size. I will talk about that in next slide. The signal for LAR is not a mini size, but because of the composition of the PLJ in the formulation present the unique challenges. So we'll talk about this in the next slide. Now, the complexity of formulation mainly comes from the complexity of the PLJ polymer itself. Now, PLJ, as the name indicates, polylactide coglycolide, there are two monomers, lactide monomers and glycolide monomers. So the ratio of the lactide glycolide, which we call simply LG ratio, determines the polymer properties. Most of the PLJs we are using has a lactide content 50% or higher, because if the lactide content is less than 50%, they do not dissolve in most organic solvent we use. So this LG ratio and also the end group can be acid or base. Not only that, lactide glycolide sequence makes the polymer sometimes dissolve in the same solvent, sometimes it does not. So molecular composition is one of the important parameters that control the PLJ behavior, also molecular weight, and the other parameters are molecular structures. Linear structures versus branch structure, the main difference is the viscosity, even if they use the same concentration of PLJ. So today, let's assume that formulation intentionally have two different molecular structures, different molecular compositions, and different molecular weights. If all those three different composition parameters are in the same formulation, furthermore, if the total amount of total amount of PLJ in terms of weight is less than 1 milligram, then we have serious challenge here, because for using conventional assay method, we need more than a milligram quantity for individual assays. So having less than 1 milligram poses serious challenges. Now, in ophthalmic product, Durista and Ozurdex have in terms of the composition, is known because the Durista contains PLJ, PLA ester, PLA acid. We don't have any further information. Ozurdex contains PLJ ester and PLJ acid. Fortunately, the manufacturer presented information that they use a resomer, both LG ratio 50-50, and molecular weight is 7,000 to 17,000. No more information. So question here is whether we can separate PLJ based on this acid end group or ester end group. So we'll talk about that first. Now, in this particular case, we modified the PLGA acid group using initially fluorescent probe, but fluorescent probe turns out to be not really fluorescent in organic solvent during GPC measurements. So after consultation with the FDA scientists, by the way, we published this particular approach already after we discussed with the great scientists at the FDA. Upon suggestion by the FDA, we turned our attention to UV probe because it doesn't affect by the organic solvent. So we use a pyrene methylamine to modify the acid group, and then we can measure the acid signal by measuring UV signal, and then total amount of PLJ can be measured by refractive index. From there, we can calculate total acid number. Now, total acid number is usually described by milligram potassium hydroxide, but it is not soluble in organic solvents. So we use tetrabutyl ammonium hydroxide and later convert it to the equivalent amount of the potassium hydroxide. So that's how we calculate the total acid number. In terms of percentage, how much PLJ acid is present in Ozurdex? By the way, Ozurdex itself was not available for research use because the manufacturer did not sell for research. So instead, we used Ozurdex similar, exactly the same method that Professor Feng Zhang prepared in his lab. So we use it, and then when we assay the amount of acid, it turns out to be 67% plus minus 23%. So it's close to 75%. He added in when he made the formulation. Now, the large variation is due to the use of extremely small sample quantity. That's why whenever we pick up small amount, it may have some variation. But overall, if you measure enough time, the average becomes the same as the original 75%. Now, let me talk briefly about this signal for LAR. That is, again, not a mini-size formulation, but it has two PLJ. One is star-shaped, the other is linear. And also, both of them has LG ratio close to 50-50. One is 50-50, the other is between 50-50 and 60-40. Very similar. So our job was to find a way to separate them and quantify them. So to do that, let me introduce the concept of semi-solvent approach. The semi-solvent is basically solvent that dissolve a certain PLJ, but not all of them. So for example, dichloromethane dissolve all PLJ at any LG ratios. But let's go to, for example, toluene. By the way, this blue rectangles means that the polymers dissolve in that particular solvent. White means polymer does not dissolve in the same solvent. So if you look at toluene, LG ratio 50-50 all the way to 65-35, they do not dissolve. But above 70-30 and all the way to 110, they dissolve. So basically, if the two polymer does not dissolve, one does dissolve in toluene, the other does not dissolve in toluene, basically two polymers are different polymers. So even though the name PLJ is same, based on LG ratio, they have a totally different properties. So on the right side of the table, for example, let me show you 56. This means transition lactide percent, 51% all the way to 99%. Now, in this particular example, benzyl alcohol has a transition lactide percent 56%, which means that if the LG lactide content is 56% or higher then they dissolve in benzyl alcohol. But if it's lower than 56%, the PLJ does not dissolve in benzyl alcohol. So there are different solvents that we can choose. So ethyl lactate and benzyl alcohol, two semi-solvent were the one we used to separate the two PLJ present signal for LAR. Once again, signal for LAR was not available for research, so we made in our lab based on the information in the patent. Now, this slide is very complicated because so many numbers, but my main point here is in the figure in the center here. If we look at linear polymer, LG ratio is 5149. That's the average of every single PLJ polymer in the sample. But if you look at individual PLJ molecule, their LG ratio may be different. So I just want to point out that LG ratio, when you look at from the brochure or catalog, they are average of every single PLJ molecule. So individual molecule may be heterogeneous. So if we mix the two and separate, for example, using certain semi-solvent, in this case, light gray method, gray area, then basically, what we are separating is based on LG ratios. Of course, the molecular weight also affect the role of here, but semi-solvent is mainly for LG ratios. So we can separate the two different samples based on LG ratios. Now, once we separate them, we can further characterize using GPC4D. Basically, we can calculate the molecular weight, absolute molecular weight from light scattering or intrinsic viscosity, and so on. Now, during the process, we realized that benzyl alcohol somehow interferes with the measurement of LG ratio and molecular weight. We're talking about trivial quantity in a few percent, and that small residual benzyl alcohol affect the results. So we had to find a way to remove every single molecule of benzyl alcohol. After several trials, and we found out that once we dissolve PLJ sample in benzyl alcohol, for example, we separate soluble fraction and insoluble fraction, and then go through precipitation, dissolution, precipitation again. That process at least twice, then we remove most of the benzyl alcohol to the negligible level. So the best precipitant we found was ethanol water mixture, 1-to-1 ratio. So in this case, again, the slightly complex, but the point is that we use three different PLJs mixed together. CB003 is a linear PLJ. The other one's PLJ glucose. Obviously, glucose has a branching number. And 6-arm is a really true branch stop polymer. Every single 6-arm is branched with a PLJ molecule. So once we mix these two, for example, on the right side bottom table, if we mix linear and glucose PLJ, linear and star-shaped 6-arm PLJ, 1-to-1 mixture, we can separate using benzyl alcohol and other semi-solvent. Then we can characterize this weight average molecular weight that is close to original value, but they are different because we're separating the two different PLJs from the mixture. So basically what it tells us that we can compare brand name product or any other reference product using with the potential genetic counterpart or another formulation you've made. We can compare the two, whether they are made of a PLJ similar or not. In terms of a PLJ properties like LG ratios, molecular weight, and molecular structures, and so we can quantify them too. So that's why this particular approach allows us to compare two different formulations, whether they're similar or not. The question of what makes it similar, what makes it not similar, is up to the FDA. But basically what we present is that approach to compare the composition of the two formulations, whether they consist of similar PLJs or not. So in conclusion, today I just want to mention that we can assay the contents of acid end group or S end group, even if they are mixed and the LG ratio is similar, molecular weight is similar. So we also present here today the signal for LAR. We can separate the star-shaped PLJ glucose and linear PLJs based on semi-solvent approach. Now our remaining job is to further characterize the signal for LAR based on exact quantity of the star-shaped or linear PLJ. First of all, I just want to emphasize here that when we hear glucose PLJ star-shaped polymer, please understand that the branching number is not 4 or 5. It is usually almost always less than 3. So it's somewhere between 2 and 3. So glucose PLJ branching number 2.5, for example, means that most of them are not branched PLJ. There are lots of linear PLJ too. So that is a reality. That's why, if you compare the raw materials after you make a final formulation composition will be very different. This is why I think FDA is emphasizing analysis of the formulation of the final product, not the initial raw material. In any case, we further continue work with the FDA scientists, and then our remaining job is to assay Durista. We have the capability and technology to assay all this, but simply the fact that total weight is less than 100 micrograms present another challenge, and that's what we are approaching right now. So if you have any questions, I'll be happy to answer during the discussion section. Thank you very much. I will stop sharing now.
Good? Okay, great. To continue on the intravitreal implant train, let's welcome Dr. Coleman Johnson.
Okay, thank you, Billy, and hello, everyone. The research I'm presenting today was basically a case study in reverse engineering for a complex drug product called Ozurdex. Ozurdex is a long-acting intravitreal implant based on dexamethasone and PLGA. This research was also an investigation into the critical material attributes of PLGA for this application. PLGA is a complex excipient, like Dr. Park just showed us, very complex excipient that introduces many interesting bioequivalence challenges. This project was performed at UT Austin while I was a graduate student there. Oh, whoops. Oh, great. Okay. This presentation reflects my views and others from UT Austin and should not be construed to represent the views or policies of the FDA. Ozurdex is one of many PLGA-based products that do not have a generic equivalent. There are 25, or actually Dr. Park showed 27. Not sure how that happened. There are 27 approved PLGA-based drug products going all the way back to 1989, but there are only 4 approved generic PLGA products. This is partially due to the complexity of PLGA and resulting bioequivalence challenges. So here is the Ozurdex implant, and here is the applicator for the intravitreal injection of Ozurdex. Over the last 6 years, we researched this topic at UT Austin. This was a collaboration with the FDA via a research contract, which was titled Effect of Repeat Unit Ordering on the Properties of Melt-Extruded PLGA-Based Long-Acting Implants. The key term here is repeat unit ordering. This refers to the fact that PLGA is a copolymer, and the sequence of monomer units in the polymer chain has a significant effect on the behavior of PLGA. So this contract went to my advisor, Dr. Fang Zhang, and another faculty, Dr. Nate Lind. So my advisor was in the pharmaceutics department, and then Lind is in the chemical engineering department. So that's our background. Ultimately, we published 11 articles on this topic, and my presentation today will cover our first 4 articles on the reverse engineering of Ozurdex, as well as our most recent article on a rabbit study we performed with our reverse engineered implants. So our strategy for the reverse engineering of Ozurdex had 3 parts. Part 1 was to understand the formulation, which is basically a monolithic cylinder with dexamethasone crystals dispersed in a PLGA matrix. Step 2 of our reverse engineering project was to master the implant manufacturing process, which is melt extrusion. And then part 3 was to understand the drug release mechanism of these implants and relate what we understood about the release mechanism back to the formulation and back to the manufacturing process. Regarding formulation, here's some basic information on PLGA. It's a very common biodegradable polymeric excipient for long-acting drug delivery. It's insoluble in water, but it becomes soluble and dissolves by hydrolytic degradation. This is the basis of PLGA being biodegradable. The hydrolysis process is depicted here. The linkage between the monomer units in the polymer is hydrolyzed, which ultimately leads to the reducing the polymer to the monomers themselves, lactic acid and glycolic acid. And those are naturally present in the body, so that's the biodegradability aspect. In Ozurdex, drug release is congruent with that PLGA dissolution process. This is the basis of the long-acting aspect of drug delivery because the PLGA dissolution process takes a long time. More on the formulation. From the Ozurdex patents, we learned that the implant is 60% dexamethasone, 30% of a particular PLGA grade from Evonik, which is 502H, and then 10% of the implant is another PLGA grade from Evonik, which is 502. So the difference in these is the acid end capping. The patent did indeed reveal the exact manufacturer and grade of these PLGAs. Moving on to manufacturing. We manufacture these implants by a double extrusion process. We developed this process based on the Ozurdex patent as well as our general knowledge of extrusion. And ultimately, the process that we developed worked very, very well. Basically, the process is to feed the blend of the drug and PLGA powders into a twin screw extruder. So this photo shows what the inside of the extruder looks like. So that blend is fed into the extruder. The extrusion is run at around 100 degrees Celsius, which is hot enough to melt the PLGA, but the dexamethasone does not melt. It remains crystalline throughout the process. So that mixture is then pushed through a circular die, and then the extrudate melt is drawn to the target diameter using a puller wheel. And then the diameter of the extrudate is measured in real time by a laser micrometer directly next to the puller. Simultaneously, we measure the torque from the extruder in real time. So that's really critical to the process control, which I'll explain in the next slide. Measure the torque and diameter in real time. And then finally, once the extrudate is cooled, it's cut to the correct size. The process control is very critical for the quality of this product. This chart basically shows the target operating window for achieving the correct dose in the implant based on the implant's diameter and the implant's porosity, right? So the implant is cut to a particular size. The extrudate is cut to a particular size to get the final implant. And so in order to have the target dose of, I think, 700 micrograms of dexamethasone, the implant diameter has to be very well controlled, and the implant porosity. So this target operating window shows you that you have a very narrow range of acceptable diameters and a very narrow range of acceptable porosities that you need to get that target dose. Another big factor here on the process control is that the implant has to be have a smooth surface and also have a fairly regular diameter over actually an extremely consistent diameter over the whole length of the implant, such that it can slide out of the Ozurdex applicator correctly during the intravitreal injection. So let me go back to the previous slide and mention this again. So the key to the process control is measuring the torque and having a consistent torque in the extruder, because that leads to consistent melt pressure, and then the melt can flow through the die at a consistent rate, meaning that it's then pulled to the correct diameter at a consistent rate. So yeah, that's the essence of this process, is achieving a steady state. So here's a video of the extrusion process. Okay. So yeah, here's the extruder, and then now we're going over to the feeders. Oh shoot, okay. So there, the feeder is in fact running. It's a feed rate of about 5 grams per hour, which seems very slow, but that's actually a perfectly appropriate feed rate. The dosage size is about 1 milligram. So a feed rate of 5 grams per hour is about 5,000 doses per hour, so that's perfectly sufficient. So here's the melt coming out of the die, and then it comes onto that steel plate called the guide plate. It goes through the laser micrometer, where we're collecting our real-time diameter data, and then that's the puller wheel shown there. And then finally, this is the extrudate. So at this point, we just have to cut it to the correct size. So one thing I haven't explained yet is that there's actually 2 extrusions total in this process. So we just saw a video of one of those extrusions. And 2 extrusions are required for the following reason. Starting with the drug and PLGA powder blend, it's a very light and fluffy powder, which doesn't flow very well, meaning that the feeding process is not terribly consistent. So in the first extrusion, we force this fluffy powder through the extruder, and then take that extrudate from the first run and mill it into granules. So these large granules have a much larger particle size and have much better flow. Then we do a second extrusion using that milled material, and that milled material has excellent flow properties, so the feeding works great, and as a result, the extrusion process is very consistent, and then it's possible to have excellent control on the implant's diameter and other properties. We got this double extrusion technique from the Ozurdex patent, and that's good because it means that any extent of PLGA degradation that occurs during extrusion in our process will hopefully be quite similar to whatever degradation occurs in Ozurdex itself. So to summarize the key takeaways from the extrusion process, it's critical to have a consistent feed rate. This leads to a consistent extruder degree of fill, a consistent extruder torque and melt pressure. This leads to a consistent flow rate of melt through the die, and that leads to a consistent and on-target implant diameter and porosity. Yeah, the porosity comes in because the pressure in the extruder has an effect on the porosity. So then these charts on the left show the extruder torque and the implant diameter in a process where there's pore feeding, such as in that first extrusion with the fluffy powders. And this chart on the right shows the extruder torque and implant diameter when you run the process with good feeding. So you can see that the torque is very level, the implant diameter is very level, and within that narrow target that we showed earlier. Here's how our finished implants stacked up next to Ozurdex. So for all of the critical quality attributes that we identified, everything looked great in comparison. So we used micro CT to analyze the porosity. With that technique, we can measure the pore size distribution, as well as the radial distribution of pores. You can see that, that there are more pores toward the outer, there's fewer pores in the center of the implant. By SEM, looked at the surface morphology. The drug remains in the same crystalline state. It doesn't melt, so you'd expect that the starting form and the form in the final product are the same. PLGA molecular weight in the finished product was the same as in Ozurdex. And then I already discussed porosity and diameter and porosity. The in vitro release looked great as well. These are the in vitro release profiles. In red is Ozurdex itself. We tested the release in-house of Ozurdex using the exact same method. Blue is our implants, and then the black line is Ozurdex data from literature. So they're very, very similar. The in vitro release method here, I'm going to get into later, but the basic details are the implant is placed into a 30 milliliter vial of a unbuffered aqueous medium. It's 30 milliliters of a saline solution, and the vial is shaken continuously, and there are samples taken periodically. So let me loop back to the FDA contract for this whole program. So the contract was titled Effective Repeat Unit Ordering on the Properties of Melt Extruded PLGA-Based Long-Acting Implants. And the repeat unit ordering is also termed blockiness. And blockiness can be very difficult to control during the PLGA synthesis process. During the PLGA synthesis, they start with the monomers, which are actually the dimers, lactide and glycolide. And glycolide is actually a little more reactive than lactide. What this means is that early on during the synthesis reaction, the glycolide is consumed faster, meaning that the growing polymer chains has more glycolide units, so more GGG units in the polymer chain. But then as the synthesis process continues, the glycolide is less abundant in the reaction vessel. There's more lactide now. And so later on in the synthesis reaction, you get more lactide, so you get LLL blocks. And this matters because it affects the PLGA degradation process. The glycolide linkages, GG linkages, are hydrolyzed faster. So if you have a highly blocky PLGA with lots of GG, then those will be hydrolyzed faster, the PLGA will dissolve faster, and the drug release will be faster. In the context of generics, this is important because you could have two different PLGA batches that appear to have the same bulk composition, but they actually have different blockiness and thus different degradation and drug release behavior. Therefore, PLGAs from different suppliers could have the same bulk composition, and they could appear the same by standard analytical measurements, but they could actually have different performances due to different blockinesses. So we wanted to explore this in the context of Ozurdex. We manufactured 4 batches of our reverse engineered formulation using the process that I explained earlier. These 4 batches were the same in terms of every meaningful variable, except we used different sources of PLGA in each batch. So for the 502H, which is the primary PLGA excipient, we used 4 different batches of PLGA, 2 from Evonik, which is the same material as in the brand name, and 2 batches of PLGA that were custom synthesized by Kinam to be slightly different in these properties. So Kinam specially engineered these PLGAs to be slightly different in blockiness. They're also different in some other properties, such as the residual monomer content. And then we tested the 4 batches for all of the critical quality attributes that I discussed earlier. They looked the same. The key difference was just the PLGA, the initial PLGA used to manufacture the 4 batches. Here are the in vitro release data for these 4 batches. And the result here depended on the IVRT methodology that we used. We started with the unbuffered saline release medium. And we saw that the release profiles of all 4 of these batches were not discernibly different. Also, the release lasted about 1 month. And then when we tested these 4 batches in a buffered medium, phosphate buffer at pH 7, the release profiles were different, and then also the release profiles lasted a lot longer. So this led to 2 questions. Why were the release profiles different in PBS? And 2, is PBS actually more biorelevant? The mammalian vitreous is buffered. So at face value, the PBS-based IVRT, the buffered IVRT, face value is more biorelevant. But we're not certain that this was actually the case. So that was something that we had to explore the rabbit study. Question 1, here's the answer. Looking at implant E2, which had the Evonik material, and implant formulation A1, which had the Kinam material, the A1 formulation had this earlier initial burst release. And the reason why is that the implant physically split open rather than slowly eroding, which is the typical degradation process. So this A1 formulation had this interesting behavior where it split open at an early time point, which we think was due to very minor differences in the residual monomer content of the PLGA used there. So then question 2, is this methodology actually more biorelevant, or is it overly discriminatory? And that question had to be answered with a rabbit study. We took these 2 formulations, our most interesting formulations, the E2 and the A1, and we injected them into the vitreous of 36 rabbits. This was a paired study, which was discussed a little yesterday. So in the pairing study, some of the E2 formulations went into the left eye of all these rabbits. The other formulation went into the right eye of all these rabbits. And by that technique, you can eliminate a lot of the intersubject variability and then have higher statistical power, greater discriminating power. So here's a video of the intravitreal injection process. This process was developed in collaboration with the ophthalmologists at the medical school at University of Texas, as well as the veterinary surgeons at the University of Texas. So basically what we're doing here, this is actually a infant eyelid speculum. The ophthalmologist said, you should get a speculum for this process. And we got one. We got the regular human-sized one that was too big. So we got this infant-sized speculum. And then we used the applicator of Ozurdex itself. We got those from the ophthalmology clinic at the University of Texas. And we put our implants inside of those, sterilized the whole thing, and did the injection. So the key here with the injection is the implant needs to go into the very middle of the eye. It's a small self-healing wound. And as you can see, this whole process takes about 30 seconds. The rabbit obviously was under a lot of anesthesia, but they do this in humans on awake patients, just under local anesthesia, remarkably. And then to assess the drug release, the rabbits were euthanized at 6 points after the injection. We collected the globe, froze them such that they could be dissected, and we recovered the implant fragment. So we had implant fragments at 6 time points after injection. We were able to use that to see how much drug had been released at those time points. We also took samples of the vitreous and aqueous fluids. And the euthanasia was necessary because it's not possible to safely sample the vitreous on a live animal or a person. So these are the results of the animal study. Looking at the in vivo release profiles, we saw that our 2 formulations had very similar release profiles. Also, the release profiles were quite similar, both in the shape and the total duration of release, to the unbuffered saline IVRT. Also, our implants looked very similar to the Ozurdex. Then here on the right, here's the drug concentrations in the rabbit vitreous and aqueous. Same results. Our formulations were similar to one another. And actually here, they're a little bit different from Ozurdex in these concentrations. And so if you go to the article, there's a little bit more explanation on possible reasons for this difference. as well as some explanations for why the saline method actually turned out to be more biopredictive than the buffered method. So that was a very interesting result. And here's the pairing analysis where you look at the two eyes of the individual rabbit in the same analysis. To summarize, these are the major takeaways from this program. First, the Ozurdex formulation and manufacturing process were successfully reverse engineered. Two, small batch-to-batch variants in PLGA properties did not affect the rabbit in vivo drug release, at least to the extent of PLGA differences that we evaluated. Third, the unbuffered saline in vitro release method was actually quite biopredictive relative to the rabbit in vivo performance. One of my major questions is how does this relate to monkey or human performance? That's an unanswered question. And then the final conclusion was that this research may assist the development of generic Ozurdex as well as other complex PLGA-based products. Wanted to thank everybody at the FDA, Yan, Ben, Xiaoming, Qi, and Young and Billy, as well as everybody that worked at this at UT, DigiM for the micro CT, and Akina for those specially synthesized PLGAs. So, thank you.
Hello. Well, I want to say thank you first off for having me here to be able to present. I know some of you probably are familiar with me and familiar with DigiM, though I'd like to first and foremost thank the CRCG coordinators for providing me with what I consider to be the best time slot for speaking right after the morning coffee boost. So I hope to live up to that potential. So there's been a lot of great talks so far, and I want to emphasize kind of a shift in tone here a bit, which is the previous discussions by Dr. Park, Dr. VandenBerg, and Dr. Johnson focused a lot on the kind of process development and what goes into the recipe, so to speak, of making these insertable ocular products. What I'm going to focus on is a little bit more a shift back towards some of the discussions yesterday. So this is going to be overcoming development and regulatory challenges of ophthalmic long-acting products utilizing image-based in vitro in silico release prediction. So what I'm showing here in the lovely spinning images down here are Ozurdex RLD on the left, and then the in-house Q1, Q2 equivalent that we were fortunate enough to be able to collaborate with Coleman and Professor Jhon at UT Austin over the past few months. So in general, coming from the perspective more of the generic side of it, and what does it take to actually bring one of these generic products to market, comes down to the idea of linking product design and manufacturing with performance and quality by design. So what this looks like is you start with your initial formulation, you pick whatever therapeutic you want to create a generic of, you have your molecule, you have your excipient, your manufacturing method, and then you move on to the development, so specific granulation method. Coleman did a wonderful job of highlighting some of the challenges that goes into that with Ozurdex. You extrude twice and then lead to that. Finally, you have your preclinical testing, your physicochemical, your DSC, your FTIR, as Michael was highlighting, in vitro release, and potentially in vivo release if you can. And then finally, BE studies in this case, where we've highlighted a lot of times that in the case of ophthalmics, a true BE study that might be done for oral solid is really not on the table. And so circling back to this, we can take a look then from the perspective of somebody developing a generic here, is we have our two different implants here. One made the exact same formulation, say 30% API, 70% PLGA. The polymer behavior doesn't always adhere to expectations. There can be major variations that occur with small changes such as vendor or grade. And you wind up with release profiles that look maybe something like this, where you have nominally two identical features, two identical implants that have wildly different downstream performances. Now, in the line of a lot of the discussions yesterday, you might argue that you just had a highly discriminatory in vitro release testing method, which is fair. That could be the case. But if you get to this point when you're in your quality control development of these, you don't want to sit there and say, do we have to go, how much more effort do we have to go into developing this method in order to make it relevant for downstream performance. So you also look at the time scale here. We're talking days to weeks to months to years. You don't always have a lot of time and money in order to make sure that you hit that development appropriately. So what can we do here? We have to start taking a look at mechanistically understanding what produces the variability and how can we properly account for it. So I'm going to kind of highlight exactly, and I'm very thankful again that Michael and Coleman were presenting before me, because I'm going to cover exactly those two product forms for the I have no idea how to pronounce that in a very quick way, but the intracanicular implant for dexamethasone release, and that's what I'm showing here. The RLD is, brand name is DEXTENZA, and what I'm showing on the left here is an example of a kind of the demonstration of some of the features of it. So, whoops. So with the manufacturer who makes it, the API, the dosing amount, the excipients, even this is through a database that I'll go over in more detail later, but it also has links to kind of any product specific guidances and the Orange Book application number. So as was presented earlier, this is composed of polyethylene glycol based polymer designed to swell. It's supposed to be therapeutically active for at least 30 days. And the mechanism of the insert results in a very uniquely controlled release that is almost entirely driven by the polymer properties. And more so than that, it's not just the polymer properties, because as we saw earlier, you can have the same exact polymer and have wildly different behavior. I can have a house cat and I can also have a lion. They're both cats, but they're going to behave wildly differently. So we need to have more of a idea than just looking at Q1 and Q2 equivalents here. And that's where we start coming in with this idea of the Q3. And in these cases, the exact structural equivalence is what we're looking at here. So what does the final drug product form look like? So what I'm showing here is on the left, we have the RLD, and on the right, we have an in-house formulation of DEXTENZA, where one thing that it's not immediately apparent, and I apologize, I realize my scale bars could use some work. The RLD comes in, as was pointed out earlier, at around 0.5 millimeter in diameter. This in-house product came in at around 10 millimeters in diameter, significantly larger, and it will likely have an impact on it. But there's some other things that are going on here. Is if you look on the top row shows looking down the main axis of the insert. If you're looking on the bottom row of these movies, what you're looking at is a side view, as if we cut and sweep through. And you'll notice that in the case of the RLD, there's the pores that you see on the top actually are these long scars that exist all the way along the axis of the implant that are not seen in the in-house formulation. So now we have a very interesting scenario. If these behave using through kind of monolithic diffusion or any kind of diffusion limited process, then these pores are actually going to matter quite significantly when it comes to the overall release. They provide a highway for any dexamethasone to rapidly escape through. Whereas in the in-house one, you have significantly fewer pores, so you don't have any of these highways to diffuse along, and you also have a longer diffusion path to go across. So will this have a significant impact on release? Porosity is arguably, in every case, a critical quality attribute, but is it the most critical attribute? And in the case of DEXTENZA, we have a very interesting case. So this is data provided that was initially done by the FDA and OPQ on what I'll refer to as an omnidirectional release. So this was done using a standard kind of dissolution protocol where the in-house shows significantly significantly slower release, while the RLD DEXTENZA is showing pretty fast release, almost completely done after a few days. So these differences are large, and you could argue that it might be a result of those pores that we're seeing. But as we know, DEXTENZA swells. It swells out radially, so do those pores maintain themselves there? And also significantly, if it swells out radially and we're doing an omnidirectional in vitro release like this, the diffusion pathway is going to be dominated by diffusion along the radial axis of the implant here. Whereas inside the tear duct, it's going to very likely not want to diffuse along those ways because of the incredibly small amount of room it has to diffuse out through those sides there. So that leads to the question of how can we determine what is the critical difference here? This structure, this chemistry, or potentially some other critical quality attribute? And that's where the title of the talk comes in. At DigiM, we focus on image analysis and advanced imaging, but we go a step further. We also focus on leveraging that imaging as a tool to simulate the drug release here. So on the left, I'm showing some of the structural critical quality attributes, such as the API particle size distribution. We see that varying between the in-house and the RLD. We have also the radial distributions on the bottom there, where we see some interesting fluctuations occurring in the RLD, which are almost where the API is located, which are directly correlated to where those long scars are. And on the right shows how we simulate drug release. And there's a three-step process. I'm not going to go over all the details of it, but we first find out all of the different diffusion pathways through a percolation simulation. We then simulate the changing diffusion coefficient based off of that geometry. And then using an appropriate physical model, we do a time conversion. In this case, the physical release mechanism that we figured was most likely the case here, given the polymer properties and the drug properties, was monolithic diffusion. So what this kind of this repeating video is showing is what monolithic diffusion looks like in this case, where the yellow shows where the API are, and as time progresses, you see it becomes more and more depleted as you go outward. So the release front progresses inward. So we wanted to mimic this in the case of the in vitro release, since the in vitro release, if it's omnidirectional, is going to be going along all of those different directions. And that's, in fact, exactly what we saw. So on the left is that same in vitro release profile, those same in vitro release profiles, now with our simulations on it. And what we can see is that this holistic kind of approach of looking at the release modeling based off of those structural attributes accurately captures those details. And most importantly here, when it comes down to consideration of those long pores that we saw in the RLD, we didn't include the pores as a transport phase. So that means that at no point in our simulation did we include anything to do with those pores, which strongly indicates then that in the process of swelling inside of the canal, you lose, those pores likely close up and you no longer have those features to worry about. The other fascinating thing that we didn't move forward with here yet, simply because we didn't have an in vitro release method with which to compare against, was we can choose what direction we're allowing to be the source of diffusion. So in this case, we're allowing the radial direction to be the primary diffusion pathway, following the physics of it. But we can also choose to say we're going to allow only diffusion going along the main axis here, which may be more instructive and more related to what would be a biorelevant kind of simulation. So this circles back again to the question of we can have a discriminatory result based off of these CQAs and these structural attributes that we're quantifying through imaging that we can then turn into either discriminatory and biorelevant kind of modeling here without ever having to sit there and worry, are we in an appropriately buffered system? How is this relevant? What is our pH? We can always account for that in silico without ever having to change any kind of thing in the lab. So that's Dextenza. Let's turn around and now look at Ozurdex, which is, again, a dexamethasone-based implant, except this time, rather than going into the canal, as with Dextenza, it goes right into the vitreous humor. Even harder, I would argue, PK measurements you would have to make there if you wanted to make them. Nobody wants to remove their eye in a clinical study in order to measure this. So, again, showing what some of the kind of features are here. We're now in the realm of PLGA instead of polyethylene glycol, again, around 30 days. And here's where the interesting thing comes into place when it comes to these structural quality attributes is with Ozurdex, we have a different polymer, different drug loading, close-ish drug loading, I'll say. But are these things going to have the same, they're going to have the same attributes we can quantify, but are they as critical? So, in the case of Ozurdex here, I'm just looking at the RLD here because this is already a host of information that we can extract here. So Coleman presented on the differences between the in-house and the RLD, and showed how micro CT can be used to quantify the pores. What we're doing here is going not only quantifying the pores, but we're also quantifying what we refer to as the API-rich domain and the polymer-rich domain. The average size of the API that goes into Ozurdex is around 2 microns in diameter, which is a really difficult size range to hit with micro CT, especially from imaging to quantify it exactly. Not because it's hard to spot the API, but at the amount of drug loading we're looking at, the polymer gaps between the particles becomes a challenge to actually spot. They're so small that they're below the pixel limit. But in regions that have a lot of API particles versus a lot of polymer domains, we can have variation in the contrast there. And so we're able to extract out where the API likely are, where the most amount of API are, combined with the polymer and the pores. So on the bottom there, we're showing the size distribution of all 3 of those domains, and then that model showing what the implant looks like. So from there, we progress down the same pathway we did with Dextenza. We go through release simulations. And so I'm showing 2 here now, because on the left, we have we did a monolithic diffusion simulation here, which is an excellent and easy starting point because there's nice closed form analytical solutions for that. And most people have heard of Higuchi, and nobody kind of argues about what's going on there. The challenge is that none of these, like the release curve looks nothing like what compendial in vitro release methods look like, what is looked like in the product listing. So monolithic diffusion doesn't capture the in vitro release of Ozurdex, suggesting that the API size, the uniformity, and the porosity are less critical quality attributes, or less critical as quality attributes, I should say, for the release performance of Ozurdex. And then we look to the other side. We did a polymer erosion-based method, where this time we don't even look at the API beyond knowing where it is spatially located relative to the polymer-rich domains. And in this case, we do an excellent job of actually capturing those same features there, which suggests that in the case of Ozurdex, when you undergo release, the more important dominating mechanism is going to be that erosion. And we learned this from the structure combined with some insight into how these are made. So this leads us to an interesting case where we have 2 ocular implants, or inserts in this case, that have very, very different critical quality attributes. I shouldn't say very, very different, but what makes their release and their performance happen is fundamentally different. And if you follow this kind of train of thought, it kind of opens up a concerning moment in generic development of, do you have to measure everything new every single time for all of these products? Is there a way that we can avoid having to constantly keep testing new RLDs over and over and over again? And to answer that question, we at DigiM decided that we've looked at so many of these. I know a bunch of you in the room are well aware of the number of products we've looked at. So we're moving into a realm where we want to make it accessible for everyone. So we developed this website called MyQ3, and I have to admit, I always look at it and try and pronounce it Mike, but it is MyQ3. And the entire idea behind it revolves around this idea of the current Q3 structural attributes are not really, they're not listed in any kind of the patents. In the product-specific guidances, they are suggested, but again, the agency wants to take a more conservative approach, so they will not tell you exactly what quality attributes to go after, which is fine. So what we want to do then is create a reusable database that everyone can have, that can, if they need to work on the development of these things, can gain access to. And so this website is, I want to say, live. I will add the disclaimer at the bottom. It is not officially endorsed by the FDA. This is work that the FDA has consulted on and is aware of, but it is not endorsed by them. Some features presented also that I'm showing may be out of date or in development, so just bear with me on that. But the idea behind it is we now have a website and database where we keep track of all of the different products that we as DigiM have looked at. So RLDs or on-the-market generics are very much in play that can go in there. So this standardizes the microstructure critical quality attributes. It makes a reusable database so you don't have to constantly be in your quality control, be checking against your own acquisition of RLDs. It's accessible. All you really need is a subscription and a computer and an internet connection. And then it allows us to inform, de-risk, and accelerate product development. So the entire idea behind this falls in line with a lot of the goals that were laid out earlier in the year by the FDA's kind of drive, especially in the realm of GDUFA, which is how to match the performance of a reference product cost and effectively, and how can we democratize that. And this database, we believe, will actually allow for that, because now any hurdles you have in acquisition of an RLD combined with any other features that are of the testing that you're interested in knowing, case in point, Ozurdex's porosity is not impactful of its release, but its structural stability and how it gets injected into the eye. Absolutely, if you have something that's too porous, as Coleman was showing, it might have structural problems in terms of its rigidity. So the way this database works, is essentially you find a product that you're looking to test. In this case, I highlighted a microsphere. You identify the product of interest, and you can compare against all of these different features that we quantify. So API size distribution in the final product form, PLGA grade that was used in it, pore radial uniformity. You can see the actual images of the materials we've collected there. And the whole idea here is that it reduces development cost, and this technology is well backed up through patented technologies as well. So if in your totality of evidence, when you're submitting a filing, you want to be able to show how comparable your work is, you can leverage DigiM's capabilities to directly compare your product against what's in the database without ever having to necessarily buy a new RLD. I won't speak to pivotal studies where the agency will likely tell you still need to buy the RLD, but at least in the de-risking and the quality control, you no longer will have to be required necessarily, if you want to quantify these, to directly quantify them first. So with that, I will end it here and turn it back over.
All right, let's welcome for the last speaker of this session, Megha Barot, to the stage to talk about quality considerations.
Greetings, everyone. My name is Megha Barot. I'm a pharmaceutical scientist in Office of Pharmaceutical Quality, and very glad to be here. Thank you for giving me this opportunity. So today I'm going to talk about the drug product quality consideration for dexamethasone intravitreal implant. So I'm fortunate enough that some of the speakers today in the morning, they already talked a lot about Ozurdex. So that has made my job a little bit easy. But here I'm going to talk about the product-specific guidance regarding this product, which recently our agency has published, and all the quality consideration for this drug product. Starting with the brief introduction of Ozurdex. So it is a preloaded dexamethasone implant. It is a biodegradable rod-shaped implant, and it is used as a single applicator. And basically it consists of the complex polymer, and it has 2 PLGA polymers in it. They are similar, but only difference is the end group. So one end group is terminated with the ester group, and the another one is acid end group. So the name of the PLGA in the labeling PI of RLD were recently updated as per the NF monograph. So this is the formal image of the recently published draft guidance for dexamethasone intravitreal implant. So the equivalence of physicochemical characterization, the Q1, Q2, and Q3 sameness recommendation has been addressed in this guidance to help all the generic formulator to encourage them and to guide them, because as of now there are not a single generic dexamethasone implants are available. So this will streamline the process, and it will help guide the formulators in terms of how to evaluate the qualitative, quantitative, as well as Q3 sameness of the drug product. So coming to the quality review considerations. So first, we will talk about what are the quality sameness recommendations we have for the generic manufacturer. Specifically, besides the API, this product contains a complex excipient, which is PLGA. So we will talk about what all comparative characterization studies which we need to perform for PLGA. Then we're going to touch base the quantitative sameness recommendation, followed by comparative characterization studies for the finished drug product, and few more recommendations. Now, the quality sameness recommendation: to demonstrate the quality sameness, applicant must submit comparative sameness study data from at least 3 batches each of test product as well as RLD. And this includes a thorough characterization study specifically for complex excipients PLGA, in which composition ratio of PLGA mixture must be compared with RLD in order to get the sameness evaluation. So the characterization study basically should have the polymer composition, molecular weight and weight distribution, polymer architecture, and as well as to evaluate the quality characterization of the finished product, the polymer end cap analysis, inherent viscosity assessment, as well as glass transition temperature assessment is recommended. Furthermore, it is recommended that all the ANDA applicants should provide the extraction procedure of the PLGA polymer from the RLD as well as from the test product. Now, as we already know that the complexity of the polymer mixture, there is a difficulty separating this, which we have already heard this morning from Dr. Park's presentation. And he also gave some valuable insight how to, overcome those challenges. But here, as an agency, we recommend that due to the difficulty separating this PLGA polymer mixtures, it would be advisable that the applicant can characterize the polymer mixture itself from the test formulation and compare them through the RLD extracted polymer mixture to demonstrate Q1 sameness. Furthermore, sufficient validation data for PLGA comparative characterization method should be provided. And it is essential to provide justification in case if there is a difference between the formulation to just kind of support that it will not affect the comparative safety and efficacy of the drug product. Coming to the quantitative sameness recommendation, so since it is not mentioned officially, but if an inactive ingredient level exceeds those in FDA approved product for the same route and use, applicant cannot really solely rely on database citation or reference drug labeling, but must provide safety justification for higher level to meet 21 CFR requirement. Now, so far we discussed what are the comparative characterization one should do for the PLGA excipient. Now let's look at the comparative characterization studies recommendation for finished drug product. Again, this must be done for 3 lots of proposed drug product against 3 lots of reference product. So the first study, which includes the physical characterization of the finished product. So for that, the appearance, physical dimensions such as length and diameter, as well as weight of the finished implant should be compared between test as well as RLD. Furthermore, dose uniformity should be evaluated following USP 905 general chapter. After that, microstructural characterization needs to be compared between test and RLD, wherein implant inner and outer surface structure morphology can be evaluated, and the structure porosity needs to be evaluated as well. Now, the third one is mechanical characterization, in which implant breaking force needs to be compared between test and RLD to make sure that implant is not getting fragmented within the injector before even getting inside the vitreous cavity of the eye. The next is dexamethasone physical property. So API sameness needs to be performed within the implant. So for that, the recommended the tests are particle size and particle size distribution, followed by polymorphic form or crystallinity evaluation. And the final one is implant in vitro degradation profile. So for IVRT test, typically it includes 3 batches, each of test product as well as RLD, with at least 12 units of every batch. and it is recommended that IVRT method demonstrates reproducibility and discriminatory ability through comparison with intentionally varied test product having a different formulation or process parameter. So the varying material attributes, for example, API polymorphism or API particle size. Then the second is process attributes by changing the particle size distribution or by changing the formulation attributes such as drug polymer ratio. So by doing those discriminations, applicant needs to show that the provided IVRT method are specifically unique for this particular test product, and statistical methods like F2 similarity factors are suggested to establish dexamethasone release equivalence between test product and RLD. Now, so a few more general recommendations. Since this particular drug product is solid, therefore, it remains unaffected by what orientation applicant is using, but irrespectively, applicant must justify that chosen stability testing orientation with supporting data in their ANDA submission. Now next, let's talk about the packaging system. Since this is the complex product, therefore it is recommended to use 3 submission batch, which is completely packed inside the injector and the final applicator to evaluate the filling process impact on the product quality due to the complexity of this product. So usually the filling process can cause quality defects, and therefore 100% inspection and EQL testing of submission batch that replicates commercial manufacturing condition is recommended. So now next is a microbiological attributes. So since this one is the complex combination product, so in this particular case, bacterial endotoxin test should be evaluated separately for drug product as well as for the device. So for drug product, applicant can refer to USP 771, which also outlines the specification limit not more than 2 EU per dose per eye. And regarding the device, you can also follow the guidance endotoxin test and recommendation for single-use intraocular ophthalmic devices, in which the recommended limit or acceptance criteria is given as not more than 0.2 EU per device. And until and unless this endotoxin evaluation has not been performed, those applicators are not acceptable for use for intravitreal injections. Now coming to the device recommendations, so since RLD is a preloaded biodegradable single-use applicator device, it is a sterile dosage form. So implant and applicator, they both are the constituent part of this device. Therefore, it is recommended that applicants should thoroughly examine the RLD device characteristics so that they can thoroughly compare it through their test device. And they can use specific parameters, for example, implant size, dosing approach, and applicator specifications such as preload applicator specifications in order to check the sameness between the device from test as well as RLD. Now, user interface assessment, applicant should refer to the given guidance by the agency, and they should include a comprehensive comparative analysis of user interface design between the proposed generic product and RLD to demonstrate that acceptable design differences are, if there are any design differences, they are acceptable and it is not affecting clinically anyways, and it should be equivalent. So with that, I conclude my presentation, and I would like to acknowledge our colleagues in OGD as well as OPQ who have been involved in preparing this PSG guidance, as well as they are also heavily involved in answering pre-ANDA queries for this particular drug product. Thank you so much.
Okay, now for the fun part, I guess we got our panel discussion. So besides the speakers, I'd also like to welcome young Jhon. He's a senior chemist in the Division of Product Quality Assurance or Assessment. He's here, yes. So get all the speakers up here as well as our panelists. So start brewing some questions. We'll take questions both from in the room and online. And is Dr. Park on also?
Yeah, I think you disabled my video, so I cannot turn it on.
Give one of those mics a check. Perfect. Dr. Park, can you hear us?
Yes, I can.
Okay, he might show up. Oh, okay, there he is. Okay, do we have any questions from in the room? If we don't, I guess I can. Oh, we got a bite.
I have a couple of questions. The first question is regarding the polymer molecular weight, perhaps for Dr. Kinam Park. So he explained about complexity of the polymer. Okay, so one of our experiences that when we were targeting a particular polymer weight, say for example, 17,000 to 19,000. So even determination within plus or minus 1,000 molecular weight is a variation which you get. But the supplier, sometimes what happens is we were finding some differences during our development. And then when we investigated, we found that the supplier has manufactured one of the batch which was having about 16,000 molecular weight and another batch which was having about 20,000 molecular weight. And then they mixed it together to achieve that range of polymer weight. So from the suppliers, I mean, from the DMF of the excipient perspective, is this allowable or does it make any change? Theoretically, it seems that even after mixing, because it's a random copolymer anyways, even after mixing, you are going to get a right property and the right molecular weight. So what is right to do? I mean, is it okay to mix the two molecular weight, one, and second, does it have the similar properties? And can we, if we show that they have a similar property, whether that is acceptable?
Well, whether they are acceptable or not is up to the FDA, but this is another reason why FDA focuses on analysis of the final product, not the raw material. So aside from whether acceptable or not, the problem with the current commercial product is that they measure the molecular weight based on inherent viscosity. So from the very beginning, even before they mixing two different batches, the molecular weight are very heterogeneous. So mixing the two batches are even worse. So there are a variety of different molecular weight in that particular batch. And depending on which portion of the batch you select, they're going to have a very different molecular weight. So to really want to make sure that if you will really want to make sure you have a certain narrow molecular weight range, you have to isolate by using some precipitation, using certain semi-solvent, etc. That's the only way to have a narrow molecular weight. So again, the commercial products are measured by inherent viscosity, which has a large variation. They do not measure by GPC using light scattering, which gives you the absolute molecular weight, because it's time consuming and it just takes time and money. So that's why they use a viscosity measurement. So this is another reason why commercial products need to be more upfront in measuring their molecular weight. But again, that's commercial products. And that's again why you need to be very careful in comparing your potential generic product with the brand name product. You have to consider the final product.
Thank you.
I just add a little bit my 2 cents to Professor Park's in the comment, because in the general polymer science knowledge, what is the most in the effect to the physical chemical property in the polymer, and even the little bit different of the molecular weight versus the molecular weight distribution. And so your idea within the two different molecular weight polymers mixture making has in the much broader distribution. That might be the causing to the final drug product quality. And also in generic side, we just recommend for each step of the quality control can best in the quality control of the final drug product. And so if you can figure out some different of the molecular weight of the PLGA may not affect the final drug product quality, that might be acceptable. But is in the mixing and then make the complex product as make the more complex, that man isn't really hard to judge for that.
Yeah, I think that was the actual challenge because see, you are mixing something around, just a little bit lower and just a little bit higher, and therefore it is very challenging to differentiate whether it was mixed or not, right? Unless somebody tells that, yeah, it was mixed.
Yeah, and so you just in the background search how much in the range may be acceptable, that's in your portion of the work, and then you propose the appropriate justification within which range of the PLGA or other polymer excipient, and then we just saw that, and then that's reasonable, and then why not we just approve for that.
Yeah, thank you. The second question regarding the implant, yeah. Regarding the implant, how do you determine the batch size of the implant? Okay, batch number of the implant. I'm just theoretically thinking that because it's a very independent process. You see, if you can define batch by just a 50 grams mixing of the polymer and drug, and then doing 2 times of extrusion, where there is another critical process where you are grinding it. So, and overall, if you see the yield, what you take, I'm just imagining the yield would be very low. So what are those characteristics which are needed? And how do you define? Because in the guidance, we say that, okay, take 3 batches, but 3 batches of 12 units each, but then each unit itself is different. And throughout the extrusion property, even if you are controlling, essentially there will be a lot of processes which will involve the rejections of intermediate strands, which are not in the specifications and all that. So eventually, telling that, okay, this is the batch number, how does it make this one? Instead of that, can we have like number of units defined? That, okay, this much units is good enough and it is representative over perhaps prepared over 1 year or whatever. I don't know what is the right way. So that was another question.
Well, I'm not sure about the regulatory aspect of that question, but one thing I can say is that one of the main advantages of the extrusion process is that it's a continuous process. And once it's reached its steady state, you can manufacture as many units as you want. So, so defining that steady state and being within that like quality by design framework means that manufacture many doses, and then if you're within that steady state space, right? Then then the batch size can kind of vary according to your needs, right? Yeah, that's my thought on that. Is that the continuous extrusion process kind of grants a lot of flexibility.
I can maybe add a little bit more comment for the regulatory perspective. Even the extrusion process is a continuous process, but you have to once you try to within the initial API batch and to PLGA last of batch, and then you mixture, and then you just once in the steady coming out the product, and then the final product within the filling process, that's we define within the one batch process. And so after the manufacturing is another batch start within the new API source, maybe or same API source, but we just recommend in the 2 API source, and also in the excipient source, maybe you can use in the same excipient source, but at least in the 2 API sources, and then making the final, the drug product batches should be in the 3 batches. And so that's maybe the answer to your question.
Okay, thank you.
Yeah, and I think it's just to tag on to that. I think it's preferential that you try and use multiple excipient sources when the excipient is complex, but we understand availability can be an issue.
I have a question for our DigiM colleague. So the technology that you have is fascinating. I think it provides a potentially useful approach to providing some standards. And so in the context of manufacturing, have you seen it being used as in process controls? Has it been used to clear batches or for both reference products and potentially for generics as well? And how costly is that technology and adopting it in the context of batch-to-batch manufacturing analytics?
It's a really important question there. So in terms of the manufacturing and batch-to-batch variability, we've used it before, and we've been able to use it across different dosage forms as well, like from long-acting and in the ocular space, as well as other dosage forms. The utility of it on a batch-to-batch kind of application is you kind of hit the nail on the head in that it's a costly both time and monetary expense. Insofar as if you have a PAT like with the laser micrometer on an extruder, that's like a PAT technology.
Exactly.
You can very quickly make changes.
How long does your technology take to process?
Several hours to, if we're running a release simulation, to potentially up to a few days. So it's for sure an offline tool. That being said, depending on where you stand in your manufacturing, it can absolutely be used very well. We've worked in the generic space in terms of pivotal studies. We've worked in that using to kind of demonstrate the comparison between the RLD and different batches of the test products, as well as prior to that in initial kind of de-risking. So it has great utility. Just how exactly it gets applied and when has to take a little bit of extra thought on it.
Is the most of that time processing, or because technology has the potential to improve? Can you make that go faster? I know there's potential there, right?
There's some hard and soft limits. The hard limit is when we run imaging. We can only go so fast. Or for instance, micro CT, we have access to synchrotron-based technology where the speed of acquisition for is on the order of seconds, but then getting time on those facilities is a bit of a logistics headache, so it takes a lot longer there. On the other side, there is a strong analytical and processing component as well, which is entirely driven by computation speed. So, you go from one server block to a data center, and you drastically drop your time. So to that extent, yes, we can. There's some aspects that can speed up, some that still are a bit of a hard cost.
So right now it's involved in database buildup, it sounds like. So that's a good start, I guess. But
I would love to build a database. I would love to have multiple servers to run these on.
I'm looking forward to the future of that. Thank you.
Hello. So I've got two questions, one for Andrew. So in terms of maintaining your database, how do you update it, or how do you think you would update it over time? So for example, if the RLD makes a change in, say, polymer manufacturer, say purity, and it's accepted by the FDA, not that you would necessarily know that information, but is there an advantage of doing, say, like micro CT of a product over, say, 5 years, you reevaluate that? That's
a great question. If the FDA is wanting to share that information of when something changes, we wouldn't say no. But realistically, it comes down to kind of surveillance at that point. At the moment, the database is very nascent, and it's been in development for a few months, and it's been live for, I believe, a week and a half at this point. So we haven't directly come into that question yet. But realistically, the best answer to that is surveillance. So if there is a change or anything that happens where we know, for instance, God forbid, Evonik goes under for whatever reason, and then we know that a lot of products that use Evonik-based Resomer or anything, we would want to take a look at that again. So market surveillance is the best life cycle management for this database. the technologies that with which we and workflows with which we characterize are very routine for us at this moment. So it's not like we have to come up with new experiments in order to verify a lot of these things.
You want to go? Come back to me.
Okay, some online questions. The first question is to Dr. Park. So we just discussed a lot of PLGA. They just want to know a little bit more about how to determine the blockiness of the PLGA.
Yes, blockiness is in fact one of the important parameters of the PLGA characterization. The way we do measure blockiness is to measure carbon-13 NMR and compare two peaks. One is 166.41, the other is 166.33, which represent glycolide peak. The other is glycolide lactide. So we compare the ratio, and we can measure the blockiness. And the reason it's important is that when we separate PLJs based on first of a semi-solvent, it separate them by LG ratios. And then we measure by GPC4D, we can measure absolute molecular weight using light scattering. And then we can add to the two parameters, you can add the blockiness, then you can clearly see the break as certain semi-solvent range. That's where you can characterize whether there are more than one PLJs, and if so, what the LG ratios are. So the three parameters, LG ratios and blockiness and molecular weight, can give you exact information how many different PLJs are composed in a given formulation.
Can I add a little bit more? The Dr. Park isn't the, if I'm just saying maybe the incorrect or correct, just not the, if it's incorrect, just correct me. It's actually, I think in the last of his research of the LG ratio, we assumed that is in the random copolymer sequence. Because in the blocking is why is in the important, but how much important? That's in the different point of view. Because if you just think about exactly in the AB copolymer, where in the one block is totally in the glycolide and the other block is a lactide. And then even in the same LG ratio with the random versus is a blockiness, that might be affected in the solvents, the solvent effect, and also in the LADO in the physical chemical property. But how much in the blockiness is in the randomness versus in the completely block is affected in the total physical chemical properties of PLGA. That is kind of the millionaire question, but we don't have an answer, but we just try to figure out for the blockiness how much affected it. But at this moment, when I just saw the LADO in the PLGA manufacturing process, they just try to not separate out within the control of the blockiness. That means it's a kind of the random copolymer processing. That's maybe my answer for the. It's maybe the blockiness might be affected, but I think it's not much effect to what we think. But maybe I'm wrong in maybe future science research.
Jhon, that's a billion-dollar question. I think the issue is not blockiness itself. Blockiness is one parameter affecting solubility in different solvent, but also solubility depends on molecular weight. Smaller molecule dissolve better even if it's more blocky. So it's not just one parameter which is important. It's a multiple impact like LG ratio, molecular weight, blockiness, all work at the same time. That is another reason why FDA is right. You have to analyze the final formulation because the same composition from the beginning, API, PLJ, were well known. Depending on how you processing parameters ultimately affect the final formulation composition. This is why initial composition is one important aspect, but more important factors are how they are processed, what kind of solvent are used to make a microparticle, how they dried, whether they treat it with the ethanol water mixture, etc. So those are all very important parameters affecting the final formulation. That's why simply focusing on initial API, what kind of PLJ used, is important, but not as much as a whole processing. That's why, again, we need to study more about how individual parameter composition as well as processing affect the final formulation, especially nowadays, is more doable because of the AI. Obviously, AI is a very broad term, but because of the AI, again, yesterday Xiaoming also presented, because AI, we can analyze the impact, relative impact, or compare the impact of each parameter of the final formulation. So still, we don't have enough data points to train the AI, but this is a good time to start considering utilizing AI in understanding PLJ formulation even better.
If I may chime in a little bit
Sure,
here.
go ahead.
Yeah, first of all, thanks, Dr. Park. Very thankful we had the opportunity to initiate our initial collaboration now, like 13 years back. I just take a step back for blockiness, for instance. What we really started and triggered our interest is we found when we researched in situ forming implants, we had data to show on face value those polymers are the same, appear to be L-to-G ratio and molecular weight range was very similar. And all those, then the results are quite different. So we were puzzled. And then one possibility has not been well studied or in depth. It was the potential for blockiness. So that's what prompted our research efforts to look into further into that direction. And that's when we introduced or initiated a project with UT Austin. We built that to study, then also continue the collaboration with Kinam. And then we further studied also in the high extruded implants in situ forming. So then right now with all the data together, I would anchor what Jhon and Dr. Park was saying. The data actually start to show it's not always the same trend and behavior. Blockiness certainly is overlooked most of the time, but it's a critical thing. But then again, emphasize on as of now, for instance, especially if the bioequivalence is supported by a PK study, it's good to characterize your raw material for all the aspects you can, because blockiness not only affect the performance, also affect the processability, meaning your manufacturing procedure, right? If you have to dissolve inorganic to make your microsphere, it takes various time frames. So that's all those are practical things to keep in mind during development. But what I'm trying to say is if your bioequivalence is still supported by a PK study, we do recommend you to characterize comparatively for certain polymer characteristics between the ILD and the generic, but we don't expect you to really show identical or highly similar for all aspects. So for Q1 sameness, as we outline in our PSG, our general expectation is you give us the data on L-to-G ratio, the molecular weight, and architecture. But for the rest of the characteristics, those are critical things to make sure you understand your product so you can set proper spec to ensure the consistency for quality purpose. But if you see differences, it's not a deal breaker. Don't panic. And if you want more clarity, come talk to us. So that's the key I want to add here. Thank you.
All right, not to keep asking questions to Dr. Park, but to Dr. Park. So you highlight a lot of challenges in terms of looking at mini-size products. And so from your viewpoint, what do you see as the most challenging aspects in terms of that type of characterization? So you hinted at, potentially high variability in, these PLGA blends. You're going through this process of solvent dissolving and then solvent exchange to get, insoluble versus soluble PLGA portions out. Or, in terms of GPC, you're trying to analyze a very small amount. So in your viewpoint, what do you see as the most challenging aspect moving forward?
Well, I think the PLJ, first of all, I just want to emphasize again, it was the FDA who started to characterize the PLJ. Without FDA, we would not have any of these technologies. So again, that's very important that FDA went through all this process. Now, the importance of the characterization PLJ, as you said, we can characterize all this composition, we can correlate the processing parameters with the final drug release characteristic, etc. But the question is, three-dimensional structure, as DigiM mentioned here, they can measure three-dimensional structure, but Q1, Q2 is one thing. And actually, FDA doesn't want to say Q3, but structural construction of the final formulation that also affect the drug release kinetics, right? So that particular aspect, three-dimensional structure, again, because of the FDA support, we were able to study three-dimensional structure of the PLJ formulation. And that aspect need to be combined with like a DGM type analysis to finally conclude whether two formulations are similar or not. So by combining all this, we may be able to conclude whether one product is similar to the other. And I'm talking about without doing a bioavailability study. You know, all this PK study is done to show they're similar, then anybody can do that. But before that, whether we can conclude that two formulations similar, that includes, again, all the characterization of PLJ molecules in addition to the three-dimensional structural characterization. We developed technique because of FDA, but combining all this, again, I'm going back to AI. Now, AI can do a lot of things we couldn't do because our analysis is limited, but AI can do many things. But again, I want to emphasize AI cannot do all this analysis because we do not have enough good data. If we do not have a good data, AI will use garbage data and throw out the garbage outcome, right? So we need to continue to build up the data bank that we just mentioned before. We need to have a good data that AI can use to have a good analysis of the whole thing. So to answer your question, in addition to analysis of individual component, I think we need to focus more on three-dimensional structures of the PLJ molecules, how that affect the drug release profiles.
Thank you.
I'm going to just tag on to the end of this towards maybe the panel is because microstructure is so important to kind of performance, or at least has been highlighted a couple of times. Obviously, some of the more like innovative analytical techniques can be used as part of, BE or pivotal study demonstration, but what are some of the kind of techniques or the role of other kind of surrogate measurements as far as like thermal and mechanical testing towards quality control of implants? Or where do you think it kind of, what's kind of the state of microstructure characterization in that aspect?
I'll jump on that. When it comes to microstructure, it's still a nebulous enough term that a lot of these different aspects all fall under it. So at the most basic level, at almost the chemical level, what Dr. Park was describing, you have 3D structures generated by, say, your polymers growing out of it. And in the realm of blockiness, for instance, that's something that impacts the structure. It's a part of the structure of the polymer that can then generate larger kind of, like if you have a tri-block copolymer, you can get spontaneously self-assembled phases in there. And that's obviously going to have some kind of impact on it, but you might not be able to characterize that with some of these novel techniques that we at DigiM utilize, like the micro-CT or SEM, and you need something like small angle X-ray scattering, or you need something like DSC. So in the state of it, and this kind of plays into the theme of a lot of what's been discussed so far, as part of the totality of evidence, if we think of like the totality of what is the structure, your dynamic mechanical analysis, your TGA, your DSC, your micro-CT, all of that plays into a holistic understanding of the product and how it can affect it. But I think that's not a satisfactory answer when it comes to development, and it comes down to whittling to what is the most important like subset of those. Is DSC more important than like PXRD in this case? That's where it starts. The challenge really lies in the next generation, because now there's all these tools available. It's what tools are the best ones to apply.
Oh, I just add a little bit more to that. I don't know what is the best answer for the best tool for we figure out for the microstructure. And so in the quality review perspective, we don't know for some advanced and some inexpensive measurement for the generic company. And so that's what we do with just the base step by step. We just try to the control for such as in the API control and each in the excipients in the same as in, actually it's not exactly the same, but statistically same as in the two polymer excipient and the different polymer excipient, and also in the each manufacturing process during for example, in the dexamethasone, Ozurdex case has a lot of maybe manufacturing process within the temperature and speeding of the extrusion that might be affect to the final microstructure of the product. And also for the dexamethasone insert, is that Dexanja, right? And Dexanja case has in the polyethylene glycols or glycol of the cross-linking density might affect to the final and the microstructure of the final product. And so that's for the totality evidence of the approach. We just recommend to we just list of the well-known and inexpensive method to figure out within the same as in the RLD and general product. And so we not perfectly know about each microstructure is in the same as in the RLD and general, and then that how much in the difference, how much affect to the final release behavior or bioavailability, we don't know. And so we just try to at the very beginning and then step by step control. And so we just overall, all of the result within just going to the one direction within the RLD and generic product is similar, and then we accept for that.
One more thing I would like to add here is like the surface texture as well as the porosity comparison between RLD and test product will be a very beneficial tool in order to overall evaluate the microstructural similarity. Thank you.
I'll give it another tap.
Hello. I just would add to what Andrew and Jhon were saying, like the different microstructural characterization techniques, right? They're kind of like profiling from a different angle almost. And a lot of these are sort of derivatives of the same fundamental properties, which can be like the cross-linking density, and like functionalization of the polymers, molecular weight, but you're kind of seeing a different aspect. Like they might trend together, but they could diverge in some cases. Like you might have the porosity, which can impact the density, which can impact the mechanical strength, et cetera, which may or may not necessarily impact the release in the same predicted way. So it's important to have kind of a holistic coverage of all of the different possible microstructural impacts on these different things, and not just assume that they all go together. And that's why it's important to have a totality approach.
Dr. Liu.
Thank you. I would like to echo what Dr. Park just said, that we would like to have more reliable data to build a database, and then we can have more understanding, which is really feed to potential AI tools. So a big applause to DigiM for their action. I would just look at their database, at least based on the catalogs, it's very attractive. Based on our experience, and a microstructure based on CT is really powerful and gives a lot of information. The other side of view, we also make quite a bit of assumptions in terms of images. So we like to use a variety of tools to really not just based on one, a variety of tools to characterize all the properties, and then correlate to the release. Sometimes we see the properties based on the images, they're different, but the release profile eventually has no difference. So that also is a complexity that we would like to be aware of that, to really get to the end point of final product. So I'm curious for Andrew that your analysis is very fascinating based on your maps of pores, which is kind of like understandable for contrast. How did you determine the API distribution? That's my curiosity. Thank you.
Everyone's favorite answer. It depends on the product. But there's at least in micro CT, and we leverage SEM as well. The contrast mechanisms that are generated in those images are related to kind of fundamental properties. So the easiest example is in the micro CT case, is density and atomic number are the main contrast generators. So in the case of Ozurdex and Dextenza, there's a convenient fact that one, the dexamethasone is in its crystalline form, so it has a slightly higher density than it would otherwise, and it also has an extra fluorine atom. Well, it has a fluorine atom in it that, relative to the other polymers present, brightens up the contrast in it. So it makes it a little less ambiguous in identification of where the API domains are. That being said, there are definitely products out there where, especially in the case of molecularly dispersed, in particular large molecule products, where identification becomes a lot more challenging, and we have to get clever with it. And it's at those points that really what we've been discussing, like the totality of evidence. So while we might not be able to conclusively say this domain is 100% going to be API, we can then use, say, knowledge determined from other techniques to supplement some of the structures that we're seeing from the micro CT.
Great, thank you so much.
Billy, can I add something here?
Absolutely.
Yeah, I think we talked a lot about Q1, Q2, let's say Q3, just structure, etc. But we need to consider ultimately the design space. Even if some parameters may be different, if the parameter is within design space, the ultimate final formulation will have the same property. So we focus too much on, similarity of Q1, Q2, but even if they are a little bit different, as Jhon mentioned before, even if they are a little bit different, the final function may be still the same. So this is why we continue to analyze, characterize the PLJ and formulation, and give some space where even if there are some variation, if within that design space, they will have the same function. I think that's ultimately by qualified design is, right? So we have to consider the differences in PLJ formulation or some Q1, Q2, etc. But still, if they are within the same design space, they will have the same function. That's what we need to consider too. So we really focus too much on similarity, but even if they are a little bit different, they may be the same in final functions.
Yeah, so I actually have a question similar to more about this Ozurdex reverse engineering and Ozurdex release studies. So when I look at your presentation, there was clearly implants made of very different polymers that without any buffer release identically, and maybe it has to do that the pH really drops in this in vitro release media and everything is auto-catalyzed by whatever this drop in pH, versus when you go to pH 7.4, you see substantial differences, right? And it's clear that they're not Q1, Q2 equivalent because you use completely different polymers. And then you go and you make conclusions that actually in vivo release is similar to, to no buffer. So what does it tell? Is it, I mean, that's, kind of contrarian question, but what does it tell? That there is a big design space that in reality none of those parameters matter because, okay, in vivo it behaves this way. I mean, this is one extreme view. The other view, maybe just for this particular in vivo study it was not sensitive enough, but in reality it's more sensitive. There are clearly discrepancy between in vitro release and in vivo release. The mechanism might be completely different. So I just was wondering how you guys all interpret, because that's a very big finding.
Right, so let me start with saying that when analyzing the material properties of PLGA or the microstructural characteristics of any of these products, there are countless variables, and it's very hard to identify which ones are going to be the most important. And so ultimately, the in vitro release test can be quite informative in that respect, or maybe like Michael said, going back to some of the fundamental properties of the PLGA, those fundamental properties might carry through, right? But ultimately, that is the challenge is you do the in vitro release test, now have to try to go back and figure out what of those PLGA properties or microstructural properties are the most important in being responsible for that in vitro performance. And that's very difficult. And then to add to the complexity, how does in vitro release translate to in vivo release? So ultimately, it is very complicated. Yeah, yeah, no easy answer to that question, I think. Yeah.
Sorry, if Anna, I think you raised a great question. If I could just chime in here again. If you talk to our biopharm colleagues, for instance, in general, right, for QC, I'm sure all companies have heard, or sometimes for bioequivalence too. Our general recommendation is, or position for IVRT is you use buffer system because it's more controlled. But then at the same time, I think in our study, it's a classic case. Now we have strong data to indicate buffer is not always maybe the best choice, depending on what purpose you're trying to achieve. So in the other decks, I can say generally speaking, right? But in the other decks case, right now, the PSG recommends IVRT as a pivotal BE, in vitro BE study. And for this type of dosage form, there is a need or expectation as for IVRT for QC purpose as well. I can't speak on behalf of the quality, but for BE perspective, we included the publication as references in our PSG. And if company decide, for instance, to go with the water system, which have shown to be more biorelevant, at least based on the animal data, as long as they can still show the validation to demonstrate the methods for their testing is reproducible and discriminatory by using their own formulation, then I think that could fit, the purpose for support as a pivotal in vitro BE. But then as for certain ambiguity or uncertainty for the quality purpose, then that's where I feel like it could be beneficial if company come to talk to us in a PDEV meeting or pre-submission stage. So they want to hear more direct feedback from the quality discipline. But I think you asked a very good question. So I think to us in general, we see the buffer is a general expectation, but it shouldn't be a default. It's all science-based. It should be data-driven and justification-driven.
Just to be devil's advocate, right? So he shows the lack of discriminative power, right? I mean, the discriminative ability is important
So that's
for IVRT, but then at the same time, there are completely different formulation use, and there is
So
no discrimination. So
yeah, the discriminatory power.
How would you show? Yeah, I mean,
it's lacking
practical
towards
question.
the blockiness. So they have the same altitude ratio and molecular weight. The real difference, right? Coleman, you can correct and provide the details, is the blockiness. So anything else we are aware of the differences?
So I thought one of the critical differences was the difference in residual monomer content. But getting back to your original question, with these in vitro methods, we only varied one factor, which was buffering. But we're neglecting many other factors that affect the in vivo release. Both of those tests are performed in water, and the volume of the in vitro release medium, as well as the viscosity and countless other things in terms of the chemical composition of the vitreous, were not represented in the in vitro test. And so here, ultimately, I think it was a coincidence that the unbuffered method ended up being more biopredictive. Basically, the buffering is not representative of in vivo, but we're also neglecting several other factors about the in vivo release. So in this case, it just happened kind of accidentally that the saline method was more predictive, meaning that it's more useful for this application. But there's definitely significant mechanistic disconnections between our in vitro analysis and the real in vivo. So so ultimately, that in vitro test was helpful for our application. But the concern is now, if you go to a product that's slightly different, or even a different generic form that's a little bit different than what we tested, well, the in vitro test isn't actually mechanistically simulating the in vivo release. So without that mechanistic simulation, it could be that that our saline method does not capture the real behavior. So there is certainly some risks in concluding that the unbuffered method was more biopredictive.
May I just add one comment related to this topic? So I think, yeah, one of the things here, it depends on what type of study we view as gold standard, right? So I think in this case, the in vivo performance is kind of like, if we consider that as gold standards, then we need to, for example, use the in vivo standard to judge whether the in vitro method we use is, for example, it's overly sensitive or not sensitive. So in this case, if we have both methods, yeah, one reflects the in vivo performance, one is a little bit more sensitive, we show the difference. I think we can probably reasonably make a judgment that maybe the PBS system is a little bit over discriminative, but that doesn't mean that it has no use, right? But for the BE purpose, it's probably the saline is good enough, but the PBS may be overly sensitive. Yeah, that's my 2 cents.
It's like fit for purpose, right?
Yeah.
Is it?
That depends on the purpose of the
exactly what
measurement.
the purposes of the measurement is.
Exactly.
It's like noodles. Do you want them hand pulled or extruded, right? And as long as they are delicious, they're good. And which reminds me, it's almost lunchtime. Thank you.
Yeah.
I think I got the answer by two of the FD persons. But to extend to that, I mean, I just wanted to know that how much is, I mean, what is please reflect about the short or a QC release method, in vitro release method, because we are right now debating about 42 days release and the differentiation. But then if, as a generic, if you want to have a short QC analysis, maybe 7 days or 36 hours, now whether that would be acceptable and what we should keep in mind. The same standard differentiating requirements, but at the same time, we also saw that those differentiating requirements are all actually not differentiating. So how do we actually develop a short-term release, 7-day release or a 3-day release? So any reflection on that?
Well, one important concept here is that in vitro release testing should be fit for purpose. And also there's this inherent conflict, which is that the more information you might be able to glean from the in vitro release test, the longer and more discriminating it is, then also it becomes increasingly inconvenient, right? So if the in vitro method is more biorelevant in terms of replicating the vitreous viscosity, for example, or you run the test for longer, then it's harder and harder to have a reproducible and convenient method. So, so the in vitro test is always fit for purpose, and it should answer a particular question, right? So if attempting to go to a 3 or a 7-day in vitro release test, it will be less informative, and maybe that's appropriate for a given purpose, or maybe it's not. So I think that would be something that some experts would have to evaluate.
Okay. Oh, got one more.
So I just had a question.
So on the same question, so if we could able to develop the short duration method with the same discrimination power, so that will be okay for agency to have in a QC setting?
Typically, if you're submitting a accelerated release test, you're also submitting a real-time release test. And both need to have method validation, which includes robustness and discrimination.
Yeah, definitely, this will qualify the method validations and robustness studies.
Yeah.
So because the 42 days for the batch release is quite big, and it will not be tolerable in the industry setting. Actually.
Right. Yeah, I think, and obviously one of the OGD people can correct me, but I think in general, it is reasonable, especially in terms of like QC, if you're doing it in vitro release for QC, as long as you have proper justification for any observed differences in discrimination or between your real-time and your accelerated, especially in terms of using it for QC. You want to demonstrate any, discrimination ability against something that would possibly happen in some kind of failure mode of manufacturing or something, or some QC or PQA that's relevant.
All right, sure, thank you very much.
So I think we've been talking a lot about design space, and my question is about the Ozurdex, for I guess the implant, because we insert it into the body. I am thinking to myself that if I am a physician, I want to know when this will completely dissolve. So for me, what would be important is the release characteristic, but also how long it takes for the whole rod to dissolve inside the eye. And so I guess we are talking about blockiness, branching, macro or micro-sized holes and pores. But I was thinking what would be important for me would be measuring how long it lasts, because it looks like it stays in there for several months. And in the study that we did, we had to sacrifice the rabbit to retrieve the rod. So I was wondering if this is something that FDA want the generic drug to measure before they approve the drug, and if so, how FDA would like it demonstrated? Do you want rabbit study? It can take several months, or some in vitro study?
So I won't answer the regulatory aspect of that question, but as far as measuring the degradation of the implant, it's actually very easy. It can just be measured by ocular imaging, and it's actually very straightforward, actually. So that's easy. You can track the implant and see it get smaller and smaller over time just by pointing a camera right through the pupil.
And also there is in the we just introduced in this morning is the two types of the drug product. One is in the Ozurdex insert and the other one is implant. Both have them has in the different duration of the time, and then where is in the administration location. And implant is a little bit longer duration, but both of them is biodegradable. And so it will be eventually gone from the body. Actually, it's a little bit different is in the conventional implant. Conventional implant is non-biodegradable.
Okay, I think we're right at noon. Oh, we got one more. One more? Sneak one more in.
Okay, thank you for that, giving me that opportunity. So I think we've heard a lot on the challenges of Q1, Q2, and it's great that the IVRT guidance has now come for the dexamethasone. But my question from an industry perspective is, looking at the challenges, if we see some subtle differences, if we see some minor differences, and we talked about design space, then do you think as a part of totality of evidence, these rabbit PK studies could be an additional tool and supplemented with modeling? Would the agency be willing to look into it, or we have to just focus on only the in vitro part and show the sameness, and this was just for your internal research, the rabbit PK studies?
I think in terms of that type of question, you should always in product development reach out through those channels. I don't think that the FDA would turn away additional information to support your package, but definitely reach out through those product development meeting cycles. So, okay, with that, I will be wrapping it up for lunch. We will return at 1 p.m. Let's thank all of the panelists. Dr. Park, thank you for also joining virtually.